Today we are going to learn in vitro methods to study the apoptotic potential of nanomaterials. In today's lecture, we are going to learn what is apoptosis and what is the difference between apoptosis and necrosis. We are also going to learn various types of in vitro apoptosis assay. At the end of the lecture, we will also have a practical demonstration to understand the apoptosis assay in detail. Let us see the difference between apoptosis and necrosis. Apoptosis is a normal or programmed cell death, and apoptosis is a physiological process, whereas necrosis is a accidental cell death and it is a pathological process. You can see the difference between apoptosis and necrosis in this picture. When a normal cell undergoes apoptosis, membrane blebbing will occur, and then the cell breaks into several apoptotic bodies which are then phagocytosed, and there is no inflammation in case of apoptosis. Whereas in case of necrosis, cell swelling happens, followed by that the plasma membrane ruptures and the cellular and nuclear lysis happens, which leads to inflammation. Most of the anti-cancer drugs kill the cancer cells through this apoptosis pathway, and some of the nanomaterials also have the potential to induce the apoptosis in the cell. In today's lecture, we are going to learn what are the various in vitro methods and assays to understand the apoptotic potential of nanomaterials. Let us see how we can detect the apoptotic cells. The apoptotic cells can be detected by analyzing their surface morphology, cell membrane integrity, mitochondrial function, nuclear events, and DNA cleavage. In today's lecture, we are going to learn this more in detail. There are several types of in vitro apoptotic assays, and in today's lecture, we are going to learn some of the important in vitro assays. Under the in vitro assay, we can broadly divide into colorimetric, microscopic, and flow cytometry and molecular analysis. Under colorimetric, we are going to learn the ELISA technique. Under the microscopic, we are going to learn acridine orange ethylene bromine staining and Hodges rhodamin staining, and also SEM analysis, how we can use the scanning electron microscope for understanding the apoptosis. Under the flow cytometer, we are going to learn the annexin PA assay as well as ROS assay. Under the molecular analysis, we are going to learn the RT-PCR. Let us learn about acridine orange ethylene bromine staining. So, this is one of the simple method for understanding the apoptosis. Here we are going to use a combination of two dyes. One is acridine orange, the other one is ethidium bromide. Acridine orange is a fluorescent DNA intercalating dye, and it is permeable to all cell nuclei and emits green fluorescence. You can see here the live cells appear green color, and when the cells undergo apoptosis, during the early stage of apoptosis, what happens is there is a changes in the cell nucleus and cell organelles, due to which the cells at early apoptotic stage appears bright green. It emits bright green fluorescence, and when we add the combination of acridine orange and ethylene bromide, when the cells are undergoing the apoptosis at the late stage, what happens is this ethylene bromide enters. You can see that here the cell membrane is intact, and here in the cell membrane is having some kind of perforation. So, due to that, this ethylene bromide stain can enter and it stains the nucleus. That leads to the combination of this green plus red gives the orange color. There will be like a orange red. So, if you are seeing the cells which are in the emitting the orange red fluorescence, that means cells are in the late apoptotic stage. And if the cell is undergoing necrosis, what happens is the cell membrane is ruptured, whereas you can see that the nucleus is intact. So, this ethylene bromide will stain the nucleus and the cell nucleus appear red color. From this data, you can clearly understand the figure A, this is a untreated, okay, and whereas the figure B, C, D, these are treated with the different concentration of nanomaterials, and you can see that the bright green fluorescence, this bright green fluorescence is the early apoptotic cells, and this orange color, as I told earlier, these are the cells at the late apoptotic stage, and here there is no necrotic cells, there is no red color fluorescence. So, when you treat with these nanomaterials, you can simply stain with this combination of these two dyes, and we can easily identify the cells which are in the early apoptotic and the late apoptotic stage. But there is one limitation in the AOEB staining, that is that staining assay can be done at the end of your experiment, and if you want to do the time dependent study, you have to go for the alternate dye, that is Hodges and rhodamin B. Let us learn about Hodges and rhodamin B staining. Here the Hodges stain, it is a membrane permeable nuclear staining dye, and that emits blue fluorescence when attached to the double stranded DNA, and this is used to differentiate the condensed pycnotic nuclei compared to the normal nuclei. And during the apoptosis, what happens is we will observe the nuclei with condensed chromatin. And we are going to use another dye, that is the rhodamin B, and this is a membrane permeable dye that stains the mitochondria and cytoplasmic compartment. You can see here the cell nucleus appear blue color and the cytoplasm appear orange color. And when the cells undergo the apoptosis, what happens is the cell, the cytoplasm will shrink, and also the nucleus will also condense, and that we are going to observe under the microscope. From this data we can clearly understand. So, this is the untreated. In the untreated cells, we can see here the orange color cytoplasm and the blue color nucleus. When the cells undergo the apoptosis, you can see that you are seeing the bright blue color. So, that is due to the chromatin condensation, and you can also see that the cytoplasm also shrinking. So, you can see that the orange color is also very less when compared to the untreated cells. So, that is due to the cytoskeleton compaction. We can also use the scanning electron microscope to understand the morphology of untreated and treated cells. You can see here the untreated cells, this is a breast cancer cell and this is a lung cancer cell. The untreated cells are like spindle shaped and well attached to the surface and intact membrane morphology. Whereas you can see that the nanocomposite treated cells, these are shrunk in size and rounded in shape, and also it is showing that membrane blebbing. You can see these are the membrane blebbing. Okay. So, these are the hallmarks of apoptotic cell death. Let us learn about the another important technique, that is ELISA. By using this ELISA, we can quantify the cellular DNA fragmentation. So, during the apoptosis, cellular DNA will be fragmented, and that can be detected and quantified by using ELISA. Here we are going to use sandwich ELISA. For that, we have to coat the microplate with the anti-DNA. That means this antibody is against the DNA. So, the DNA can bind to this antibody, and before that we have to label the DNA of the cells with the BRDU. BRDU is bromodeoxyurine. It is a thymidine analog, and this will be attached to the DNA of the cells, and once the cells are treated with the nanoparticle, the cells undergo the apoptosis. And during the apoptosis, what happens is the cellular DNA will be fragmented into small fragments, and this can be detected using the antibody specific for this DNA. And once you do the microwave, and it will convert the double stranded DNA into single stranded small DNA fragments, and we already have this antibody on the microplate, and this DNA will attach to the antibody. Then we have to add another antibody, that is anti-BRDU antibody, and which is having an enzyme called peroxidase, POD, and this will bind to the your DNA. So, as I told earlier, this is a sandwich ELISA. So, this is your microplate where you have the antibody specific for your DNA labeled with BRDU, and we are going to add another antibody, and this antibody specifically bind to the BRDU labeled DNA, and it is also carrying an enzyme called peroxidase. So, your DNA is sandwiched between the two antibodies, that is why it is called a sandwich ELISA. Then you have to add the substrate, that is the TMB, that is tetramethylbenzidine. Once you add this TMB, so this enzyme will convert this TMB into color product, and this color can be quantified using the spectrophotometer. By this way, we can quantify the apoptosis. From this slide, you can clearly understand how we can use the ELISA to quantify the cellular DNA fragmentation. And if you are getting the observance is more, that means more cellular DNA fragmentation, that means more apoptosis. Let us see another simple assay for understanding the apoptosis, that is apoptotic DNA laddering. During the apoptosis, what happens is this caspase 3 activates CAD, that is caspase activated DNA enzyme, and that leads to cleavage of chromosomal DNA. And once you isolate the genomic DNA from the untreated and the nanoparticle treated cells, and when you run this DNA in the agarose gel electrophoresis, you can observe the DNA fragments, and these DNA fragments will appear like a ladder. So, that is why it is called as DNA laddering. You can see here in the lane 2, this is a untreated one. In the untreated cells, when you isolate the genomic DNA, you are getting a single band, it is intact, the genomic DNA is intact. Whereas, in the nanoparticle treated cells, you can see that the DNA is fragmented into small pieces, and when you run in agarose gel electrophoresis, you get this laddering pattern. So, that is why it is called as DNA laddering. Let us see how we can quantify the apoptosis using flow cytometry. Flow cytometry measures the light scattering properties of cells and fluorescent emissions of molecules attached to cells. Here, once the cells are treated with the nanoparticles, then cells can be dissociated and washed and diluted using PBS, that is phosphate buffered saline. Then you can stain the cells with the suitable dye. Once the cells are stained with the suitable dye, you have to strain the cells using the cell strainer to remove the cell clump. Once you do the flow cytometry analysis, what happens is the cells which are stained with, for example, the cells are stained with the green fluorescence and some cells are stained with the red fluorescence. So, that can be detected using the detector system, and the data can be analyzed using the software in the computer. Let us learn about the principle of annexin PA staining for understanding the apoptosis using flow cytometry. The membrane phospholipid phosphatidylserine in the apoptotic cells is translocated from inner to outer leaflet of the plasma membrane. Thereby, it is exposing the PS to the external cellular environment. That means, during the apoptosis, the PS will be exposed to the external cellular environment, and when you use the annexin stain, it will specifically bind to the PS, and if this annexin is conjugated with this fluorescein isothiocyanate FITC, and it will enable the sensitive detection of cells undergoing the apoptosis by flow cytometry. In addition to this annexin, if you are adding another dye, that is a propidium iodide, it will be easy to identify the early apoptotic cells. In early apoptotic cells, what happen is, it will have the intact cell membrane. So, that will exclude the PI, whereas the membranes of the late apoptotic and dead cells are permeable to PI. So, only the late apoptotic cells and the dead cells will allow the PI to stain the cells. In this way, we can easily detect the cells which are in the early apoptosis and which are in the late apoptosis. So, from this picture you can clearly understand. So, by using this FITC conjugated annexin, we can detect the early apoptotic cells and the late apoptotic cells, whereas this PI will be helpful for understanding the late apoptotic cells and the necrotic cells. From this picture you can clearly understand that, as I told earlier, this phosphatidylserine, this PS, so in the normal live cell you can see that it is inside the cell, but during the apoptosis, during the early stage of apoptosis, you can see here this PS is exposed to the external environment, and when you add this annexin and which is conjugated with the FITC, so this early apoptotic cells will appear green color. And when you use the combination of the another dye, that is the PI propidium iodide, you can see that during the late apoptotic stage there is a membrane rupture, and due to that the PI can easily enter and it can stain the nucleus. So, when you use the combination of this annexin and PI, we can easily quantify the apoptotic cells which are in the early stage and in the late stage, and also we can quantify the necrotic cells. And when you analyze the data, you can see here, when you compare to the control, the treated one, you can see the apoptotic percentage is more, and this each dot represents the cell. So, each dot represents the cell. Most of the metal nanoparticles induce apoptosis by producing ROS, that is reactive oxygen species, that can be detected using ROS assay. We can use the flow cytometer to detect the ROS. For that, we have to use a dye called DCFH DA, which is a non-fluorescent dye. When you add the dye to the cells, and this non-fluorescent dye will be converted into another form, that is this DCFH DA will be converted into DCFH by enzyme called cellular esterase. And if there is a more ROS, then this non-fluorescent dye will be converted into DCF, that is a fluorescent. That means if you have more ROS, then you get high fluorescence. The ROS levels can be quantified by determining the percentage of fluorescent cells using a flow cytometer. Here R2 is non-fluorescent cells. We can gauge the population of cells as R2 and R3 is the fluorescent cells where ROS production is there. From this picture you can understand this is a breast cancer cell and this is a lung cancer cell, and this is treated with the folic acid conjugated nanoparticle. And when you are treating these cells with these nanomaterials, you can see this untreated one, there is no fluorescence. That means there is no ROS, no reactive oxygen species. Whereas when you treat the cells with the particular nanomaterial, you can see here the ROS production is high. So, that is the fluorescence also high. And as I already told you in one of the lecture, that is IC50. IC50 means the concentration required to inhibit 50% of the growth of cell. And when you use the 0.5 IC50, you can see the percentage of the fluorescence is less, and IC50 you are getting approximately around 24%. And when you are using the double the concentration of IC50, you can see that the fluorescent intensity also high. That means the ROS production is very high here. And from this picture you can clearly understand, we can also do the time dependent study. With respect to the time also, we can see the ROS production is high. For example, in this case, so this is a folic acid functionalized Fe3O4 nanoparticles, and that generate the reactive oxygen species. And we can see here with respect to the increase in the time, the fluorescence is increasing. That means the nanoparticle is released from the nanofiber and it is inducing more ROS. That is why you are able to see the more fluorescence. And you can also see by using the flow cytometer, we can also quantify the fluorescence. So, by using this assay, we can do the time dependent as well as the concentration dependent studies for understanding the reactive oxygen production by the nanoparticle. Let us see a simple assay for understanding the mitochondrial membrane potential. To analyze the fluctuation in the mitochondrial membrane potential, we have to use rhodamine 123. So, this is a cationic dye and it can stain the mitochondria. And during the apoptosis, early stage of apoptosis, there will be a change in the mitochondrial membrane potential. So, that can be detected by using this rhodamine 123 stain. So, you can see here in the untreated cell there is no loss of red fluorescence, the fluorescence is intact. Whereas, you can see that the nanoparticle treated cells, you can see here the significant decline in the fluorescence. So, that is due to the loss of mitochondrial membrane potential. So, this confirms that there is an induction of apoptosis after treating with the nanoparticle. So, when compared to the ROS assay, if you are having more fluorescence in this assay, that means the cells are live, the mitochondrial membrane potential is intact, whereas if there is a less fluorescence, that means the cell is undergoing apoptosis and that is why there is a loss of membrane potential. Finally, we can also confirm the apoptosis through gene expression analysis. Here we have used semi-quantitative RT-PCR for analyzing the apoptotic signaling genes, and these two genes BCL2 and BCLXL, these are anti-apoptotic genes, and here this is a untreated sample and this is a nanoparticle treated sample. You can see here in the nanoparticle treated, the gene expression is down regulated. That means these anti-apoptotic genes are down regulated, and whereas these are the apoptotic genes. So, you can see here these are upregulated. That means the nanoparticle is inducing apoptosis. Based on the various assays, we can propose the mechanism of apoptosis induced by the nanomaterial. You can see here when you are using this nanomaterial, it is inducing the ROS, so which can be detected by using this DCFH-D8 dye, and that leads to induction of P53. So, that is a P53 mediated apoptotic pathway. So, that can be studied by using the RT-PCR, and we can also understand the mitochondrial membrane potential by using this rhodamine 123 staining. And finally, what happens is, so this will trigger the caspase 3, and this caspase 3 will induce the DNA cleavage, and it breaks the DNA into small fragments, and that leads to the apoptosis of the cell. I hope you got the overall idea about the various assays for understanding the apoptotic potential of nanomaterials. Let us go to the lab and learn this technique more in detail. We have seeded 10,000 cells for the AOEB assay. Now we will view the cells under the microscope to confirm their presence in both the control wells and the wells where the test material is to be added. Next, we will place the 96 well plate in the 5% CO2 incubator for 24 hours. Yesterday we seeded the cells for the assay. Today we will prepare the desired concentration of the test material for treatment. Similar to the MTT assay which we performed in the last lecture, the test material must be diluted in the cell culture media. Once prepared, we will take the seeded cells from the incubator and treat them with the test material. Here you can see that the cells are well adhered to the 96 well plate. We have observed that the cells are adhered to the 96 well plate, and now we will treat them with the test material. Right now we are going to test the material in 3 wells for the AOEB stain and another 3 wells for Hodges staining. In this, the first and the third columns are the control wells, while the second and the fourth columns are the wells where the test material will be added. Now we will add 100 microliter of cell culture medium to the first and third column wells. Next, we will add the test material to the remaining wells. We will incubate the cells according to the treatment period, which will be 24 hours, and after incubation period, we will observe the results. For the AOEB staining experiment, we will first remove the medium from one of the control wells and add 10 microliter of AOEB stain. We will incubate it for 3 to 5 minutes and then observe the cells under the microscope for imaging. Next, observe the cells under the microscope while they are still in DPBS. We will use three filters: Trans-GFP, Texas Red and Bright Field Imaging to visualize the live and dead cells. Here you can see live cells in the control with no dead cells observed. We can now save the image. We will repeat this process for the other two control wells. After completing the control wells, we will now move on to the test wells. For each of these wells, we will remove the medium containing the treatment material and add 10 microliter of AOEB stain. The incubation time should be the same as for the control wells. In the test wells, we observe bright green fluorescence indicating early apoptosis, which suggests that the test material has the potential to induce apoptosis. We will save these images as well. We will repeat this process for the other two test wells. In this step, we will perform Hodges staining on the cells that have been seeded, similar to the AOEB staining experiment. We will begin by removing the medium from one of the control wells and adding 10 microliter of Hodges stain. Incubate the wells for 3 to 5 minutes. After the incubation, remove the dye and gently wash the well with DPBS to remove any excess stain. Ensure that the cells remain in DPBS to prevent them from drying out. Next, observe the cells under the microscope while they are still in DPBS. Hodges stain specifically binds to the DNA in the nucleus, causing it to appear blue. In this image, you can see the typical nuclear pattern indicating proper staining. This is a sign of healthy cells that are not undergoing any process such as apoptosis. We will save these images. Repeat this process for the other two control wells. Once the control wells are completed, move on to the test wells. For each test well, remove the medium containing the treatment material and add 10 microliter of Hodges stain, just as done with the control wells. The incubation time should be the same as for the control wells. In the test wells, no fluorescence will be observed in the trans GFP and Texas red channels, confirming that the Hodges dye is working properly. Additionally, we observe fragmented and condensed nuclei under the DAPI filter, which is a characteristic of apoptosis, indicating that the cells are undergoing apoptotic cell death due to the treatment. Capture and save these images as well. In these two experiments, we have performed apoptosis assay using both AOEB staining and Hodges staining methods. As a summary, in today's lecture, we learnt various in vitro methods to assess the apoptotic potential of nanomaterials. Thank you for your kind attention. I will see you in another interesting lecture.